So welcome everybody. Today is the time for the Bioexcel webinar number 85. So our speaker of today is Marco Giullini, and he will speak about modeling antibodies in the post-Alphafold area, where we are now. That's a good question. And Marco is from the Biofood Center for Biomolecular Research in Utrecht. I belong to the Utrecht University in the Netherlands. I'm Alessandra Villa, and with me there is Otto Andersson, and we are from representing Bioexcel Center of Excellence for Computational Biomolecular Research. I would like to inform everybody that this webinar is recorded. So during the webinar, you can ask questions, and to do that, you can use one of these buttons that you should have according to your Zoom application at the bottom. And then there you can type your question. You can type the question in an anonymous way or not anonymous. And it will be great if you can tell us if you have or not a microphone when you type your question, because at the end of the webinar, if you have a microphone, we will unmute you to ask directly the question to Marco, but otherwise we will read the question for you. So after the webinar, maybe you still have some question. So you could go to the Ask Bioexcel forum. Here you find the address. And since Marco webinar is really targeting ad hoc, that is one of the core applications of Bioexcel, will be great if you can put your question in the category, ad hoc category in the Ask Bioexcel forum. Here you have a QR code for the category, and then here you can see an example of the category. And Marco will be happy to answer to your question. You can also tag him for the first week. You can tag him on this forum. So something about Marco. Marco research focus on combining physics-based approach and machine learning algorithm to predict the structure and the property of biomolecular system. He did a bachelor in physics at the University of Milano, and then he moved for the master to Trento, where he did a master in quantitative and computational biology. In 2022, he got his PhD from the University of Trento under the supervision of Professor Raffaello Potestio. And he particularly was working on method development for cross-grain modeling of biological system. Exactly the same year, he moved as a postdoc in the group of Alexander Bronwen at the University of Utrecht, where he's still working on computational structural biology. In particular, is one part of his work is improving antigen structure prediction. And now I'm curious to hear what he will tell us. Please.
Hi, everyone. Thanks, Alessandra, for the very kind introduction. I hope you can see my screen and that you can hear me properly. Indeed, antibodies. This is a Bioexcel webinar entirely focused on antibodies and on how to model them in this post-Alphafold era. And here I will discuss kind of the state of the art on about the combination of machine learning approaches such as AlphaFold and more physics chemistry-based approaches to model these challenging proteins. So let me start with an overview of my presentation. We will start with an introduction about antibodies, what they are, why they are important. Then we will move to the problem of antibody modeling, and then I will introduce a new concept that we developed in Alexander's group, that is the concept of cluster diffusion ensembles to model antibody structures. And then towards the end of my talk, I will talk a bit about nanobodies, the differences and the similarities between nanobodies and antibodies and how to model them. And then, of course, I will do some conclusions. So Let's start with antibodies. They are fantastic proteins. They are extremely flexible, and they are able to bind basically any antigen molecule that is entering our body. So viruses, bacteria, they have like proteins that our immune system can recognize and that can be attacked by antibodies. And how this binding happens is that we have these molecules on the right. I hope you can see my pointer. Uh, so these antibodies are Y-shaped molecules with two different chains. Here the dark one is called the heavy chain, and the purple one is called the light chain. And you can see the heavy chain is longer, so it's also the bottom of the Y, while the light chain is only on the two tips. And what is happening here is that on the two tips of the Y, we have the variable domains of the light and the heavy chains. And on these variable domains, we have 6 loops that you can see here at the bottom, 3 on the light chain and 3 on the heavy chain. And these loops are able to bind the antigen molecules. So those loops are the amino acids that are basically responsible for binding the antigen with a high specificity. And antibodies have several applications in diagnostics and therapeutics. And at the moment, we have more than 500 monoclonal antibodies that are either approved and or under clinical investigation. So it's there is a lot of stuff happening in pharma about monoclonal antibodies, and also in terms, like in more economic terms, the market size associated to this molecule is huge. So let's see how to predict the structure of an antibody. So the problem of antibody structure prediction. In the last year, we have seen quite an improvement in the accuracy of predicted antibody structures. So before 2021, before AlphaFold, predicting the variable domain of an antibody was almost an hopeless challenge. But now the accuracy of the predicted models has increased tremendously. And indeed, AlphaFold and RosettaFold played a very important part on this. And yeah, they got the Nobel Prize a few months ago. So it's like their algorithms really revolutionized also the field of antibodies. And modeling antibodies, the prediction of the complex that these molecules form with the antigen molecule is also very important. And these two algorithms, both AlphaFold and RosettaFold, showed a substantial improvement in this task as well. But there is still some room for improvement. Some loops on these hypervariable regions are very challenging to be predicted, and still the fraction of complexes that can be modeled in a correct manner is quite low. So around 50% of the times you still get an incorrect model from AlphaFold. And even if you get let's say a correct model, maybe its structural accuracy is not great and can be improved. So the quality of the amino acids at the interface can be improved. So, but what is the factor that is making the prediction of antibody and nanobody antigen complexes so complicated? It's mostly the fact that there is no coevolution between the antibody and the antigen sequences. So coevolution is the way that AlphaFold and related machine learning-based architectures create distance restraints to basically generate the three-dimensional structure. Basically, by inspecting the coevolution signal arising from a multiple sequence alignment, so something that looks like this, a set of sequences that are related to the query. Basically, we are able to infer coevolutionary contacts between amino acids that are not in contact in the sequence. So if there is a coevolution signal, it means that the two amino acids are kind of coevolved together, and that typically means that they are close in 3D. They are close in space. And basically, AlphaFold internally uses this this context to basically set distance restraints, and with these distance restraints, then the machine learning can find a possible solution for the structure prediction. But for antibodies and antigen, this doesn't work. Between the antigen and the antibody, the coevolution doesn't exist. So it's our immune system that matures and creates the antibody to target a specific antigen, and these two guys didn't really see each other for a long time. And so the performances of AlphaFold are quite low. And you know, in the lab, we say that when your when AlphaFold is not confident about your favorite antibody antigen complex, then it's maybe a good idea to turn to physics-based prediction methods, especially if you have some knowledge about the epitope. So the epitope is the region on the antigen where the antibody is going to bind. And here you have, for example, here you have two examples of experimental knowledge about the epitope. On the left, you have a very good knowledge of the epitope, so very good epitope mapping. This is something that is not really realistic in a standard situation, but maybe at the end of an antibody design campaign, this is something you may have. And on the right, this is a more like vague definition of the epitope, in which you just you just know that the antibody is binding on a certain patch on the antigen, but nothing more than that. So if you are like in these two situations, maybe it's probably good to use Haddock to model your complex. And here I will try to explain how to use Haddock tree to perform the task of antibody antigen complex prediction. So Haddock tree, how it works for antibodies, it's quite simple. You have to start with a model of an antibody. So these days, it's very unlikely that you will find an unbound structure, so a structure of the antibody without the antigen in the PDB. So most of the times, you will have to generate the antibody with AI tools such as AlphaFold, and then of course you will have to generate the antigen as well. But for the antigen, keep in mind that it's a lot more likely that you can find a structure of your antigen, or maybe an homologous structure of your antigen on the PDB, and that is going to be most of the times more accurate than what AlphaFold is going to give you. So we don't always have to use AlphaFold to model our antigen. Just we can use it when we have, of course, no structure available. And then you plug these two molecules into what is called an Haddock tree configuration file. Basically, at the beginning, you define where the docking is going to happen, and then you plug these two molecules, antigen and antibody. Here I say antibody ensemble because it's known that for an antibody, it's better to have more models in input. And then you place a set of different modules that define your docking workflow with Haddock tree. But before going into the details of about this specific workflow, let me just spend a few words about Haddock tree, the idea behind it. Haddock tree is basically the new generation of the Haddock software, and here we decided to break away from the rigid pipeline of the Haddock 2 version, in which basically the only one possible protocol was available. Basically, we broke the code into more than 20 pieces, and now all these pieces called modules are actually can be interchanged, and the users have much more flexibility for building their custom docking workflows. For example, we have several modules that do the refinement of the docking models, different modules to do the sampling, so the real docking in which we place the two molecules together. We have a lot of ways to analyze our data, so it's much more flexible and user-friendly than the previous Haddock software. And you can easily install it with pip, and you can have a look both at the code and at the user manual at these links. So now that we know the basic concepts behind a Haddock tree, we can see how to run an Haddock tree configuration file and what is actually happening inside of it. So for an antibody antigen complex prediction, we start with the rigid body molecule, which is the real docking part. So here we generate several models according to the information that we have. And here the molecules, the antibody and the antigen, are treated as solid rocks, and they just get together, and we try to find the local minimum of the energy function. And then we have this Celetop module in which only a fraction of these models, typically 200, is selected for the flexible refinement. So the next stage, this is a molecular dynamics step in torsion angle space, in which basically both the side chains and the backbone of the amino acids at the interface are allowed to move and to find their preferred conformational arrangement at the interface itself. Of course, always minimizing the energy. Then we have a short energy minimization of the model, and we cluster them, typically using the fraction of common contacts in the models. So we want similar models to be clustered together to improve our statistics. And then at the end, we have this Caprival analysis, in which we generate all sorts of statistics and plots and data to allow you to basically pick the best, the most accurate prediction for your system. And you see here we have a scatter plot with different clusters and their score. And here you can clearly see that, you know, the score is lower for these two clusters. So if you have to decide which prediction fits better the data, it's probably going to be one of these two clusters on the right, on the left. And the same can be done on a more cluster-based manner, for example, showing the box plot distribution of the score and similar properties. So just check it out, but keep in mind that this is the way you can simply run an antibody-antigen docking protocol with Haddock-3. So we did this, and on a dataset of antibody-antigen complexes that was not part of the training set of Alphafold. And indeed, we found out how Alphafold has very low performances on this dataset. Here we're using Alphafold 2 because, yeah, Alphafold 3 didn't exist yet. So it's hard to be on par with Google. But yeah, Alphafold 2 was predicting only like 25% of the cases correctly, while Haddock, driven by decent knowledge of the interface or by a very good knowledge of the interface, is doing much better. And one thing to remember about this plot is that when you provide an antibody ensemble instead of a single antibody structure in input to your docking, then the likelihood of your prediction to be accurate is higher. So you see that there is this difference both in the true interface and in the loose interface scenario. So if you are modeling an antibody from scratch using AI tools, it's better to try to provide more diversity in the input. And just as a remark here, the Haddock protocol had a limited sampling, so it was optimized for being very fast. So if you used a standard Haddock-3 protocol with a higher sampling, probably you would get even better results. So just as an intermezzo, we have also, like, so far we discussed two cases. You know, when AlphaFold confidence is great, you should just take the AlphaFold model. That's probably going to be fine. And when AlphaFold confidence is very bad, you just want to, you know, completely ignore the AlphaFold model. But there is an intermediate situation in which the AlphaFold interface, the AlphaFold confidence is just okay. And that might mean that the interface is kind of correct, but its quality, the quality of the amino acid contacts at the interface itself may not be great. And so we use this possible solution that is basically integrating an AlphaFold prediction within a physics-based docking strategy. We used Haddock for that, and basically we use Haddock. We use the AlphaFold structure as a template for the docking. We did this for the Capri target 235. Here we have a TCR molecular T-cell receptor. Basically, it's a type of antibody, and this TCR is targeting a peptide MHC complex. And when using AlphaFold alone, we find an interface RMSD of around 4 angstrom, which is barely acceptable, let's say. And while integrating the AlphaFold structure as a template for the ad hoc docking, at the end we get a much better interface, so 1.7 interface RMSD. So we didn't benchmark this approach too much, but sometimes it can be possible that using AlphaFold as a template for more physics-based calculations is actually taking you closer to the experimental structure. Then, if we reflect a bit on what is going wrong in the cases in which ad hoc is failing, is the prediction of the H3 loop. So this loop is the most crucial one for the recognition of the antigen, and the fact that it's at the genetic level is located at the intersection between three different gene segments, the V, D, and J gene segments. Basically, it allows for a huge sequence variability in this loop, and this is reflective of course in a huge variability in the structures that this loop can adopt. And what has been proved is that the structures that this loop can adopt, most of them do not exist in what is called the non-antibody world. So whatever in the PDB that is not an antibody. So basically, we have a part of our antibody that really behaves like nothing we have seen before. And this, of course, makes its prediction very challenging. And again, I really want to remark this, even for the H3 loop, there is basically no information content that is encoded in the multiple sequence alignment. So if you look at the multiple sequence alignment given by AlphaFold for an antibody, on the left here you have the heavy chain, and on the right you have the light chain, and you can see that in correspondence of the H3 loop, typically the sequence signal is much more sparse, meaning that the sequences there are shorter and in general not so similar to the query anymore. And this lack of information, of course, is having an effect. And indeed, AlphaFold is doing much worse on this loop than with respect to what it is doing to the other in the other 5 loops. Here we can see that for the other 5 loops, so H1, H2, L1, L2, and L3, basically the average prediction given by AlphaFold has an accuracy of around 1 angstrom, which is very good. So we can say that, okay, for the other 5 loops, the problem is kind of solved. Just trust the AlphaFold prediction, and yeah, most of the times you're fine. But for the H3 loop, we can see that the average value of RMSD with respect to the experimental structure is quite high. It's almost 3 angstrom, and there are lots of cases, as you can see here, where the RMSD is higher than 4 angstrom, and higher than 4 angstrom means that the prediction is really useless. So not much you can do to save that prediction. And the good thing about AlphaFold is that when the H3 loop is complicated to be predicted, AlphaFold will kind of tell you. So if you look at the confidence, so AlphaFold has this variable called the PLDT, predicted local distance difference test, that is an amino acid wise quantity confidence metric that is encoded in each residue. And basically, if you take the average of this value over all the H3 loop, you can find that this average is quite well correlated with the actual accuracy of the predicted loop. So if you look at the right side of this scatter plot, you can see how when AlphaFold is very confident about its job, so PLDT higher than 80 more or less, then it's very rare that we have wrong predictions. So we have a couple of ones, but most of the times we have a prediction that is below 2 angstrom, while things change if we go to the other side of the plot. So when the H3 PLDT is lower than 80, you might have a few cases where the prediction is good, so RMSD around 2 angstrom, but most of the times the prediction is just very wrong, very incorrect. And at this point, you might think, oh, there must be a way to increase the AlphaFold diversity. I just want AlphaFold to generate more models for me so that I can have at least one good prediction in my sample. And here the problem is that the AlphaFold sampling, so the diversity of the models that are generated by AlphaFold is not really great. So here you have an example of the most heterogeneous conformations of the H3 loop that were sampled by AlphaFold, and you can see that they are not heterogeneous at all. So they are kind of the same structure. So if you know that that one of them is wrong, probably all the other ones are going to be wrong as well. So now we want to find a way to increase the AlphaFold diversity in correspondence of the H3 loop to, to maximize the chances of getting a good conformation on this very challenging loop. And there have been several attempts to increase the diversity in the AlphaFold output since the very early days of AlphaFold, so 2022. Several people have proposed different ideas to do this. The first attempt was the random MSA subsampling. Basically, here the idea is that you reduce the depth of the multiple sequence alignment in input to AlphaFold to be able to sample multiple conformations of your protein. And they applied this idea to a set of transporter proteins, and they found out that if you reduce the depth of the MSA by 10 or 20 times, you can sample basically both conformations. Of course, you don't have to go too sharp in this reduction, because if you go, for example, to 16 sequences only, well, you're messing up a lot in the structures, as you can see from this scatter plot. A bit more elaborate way to do this to enforce diversity in AlphaFold is through MSA clustering. So instead of random subsampling, you cluster the MSA, and you hope that different MSA clusters will correspond to distinct conformational basins in your AlphaFold sample. And the authors here tried this on metamorphic proteins, so proteins that at room temperature can change their fold dramatically, up to 1 or even 2 nanometers. And yeah, they showed that they were able to find most of the times both conformational bases corresponding to the different conformations. Another approach to enforce diversity in AlphaFold is massive fold. So here it's more like a sort of a brute force approach, in which you play with several versions of the network and different parameters, for example, activating dropout or not, or using templates. And here you try to generate just simply more models in AlphaFold, so up to 5,000, I think. And this sometimes allows you to sample conformations that were not that you could not observe with the standard AlphaFold pipeline. As you can see here, for example, the first version of the network, so AlphaFold 2.1, is working much better than version 2 or version 3. But this approach is a bit more like trying to take AlphaFold outside of its comfort zone, but AlphaFold was not really developed for this. AlphaFold was developed to predict static protein structures. So there must be a more like a fundamental way to enforce diversity in it. And I think this is something that was actually achieved by the Yakula group at MIT. They published this preprint last year about AlphaFlow, combination of AlphaFold and a diffusion algorithm, flow matching, in which basically AlphaFold was repurposed from a regression model to a generative modeling framework with the aim of obtaining a more highly diverse output while keeping the AlphaFold accuracy. And when we saw this preprint, me and my colleague Xiao Tong, we thought, oh, we should use this algorithm to sample more the conformations around the H3 loop. So to have more sampling of H3 while keeping the AlphaFold accuracy. So we tried the algorithm. Here I'm going to show you like our results. So here you see the H3 loop of the antibody 7-MSQ. In orange, you see the experimental structure of the antibody, of the loop, in which the loop is kind of folding back onto the framework of the antibody, while the AlphaFold model in blue is very shows a very elongated conformation that is kind of wiggling in the solvent. And yeah, I hope I don't have to tell you that this prediction is very wrong. There is nothing you can do to this prediction to yeah, you know, to have any reasonable answer to your questions. And we started sampling with AlphaFlow around this conformation. And at the beginning, we see very horrible models. But after a while, some models start to appear that resemble a bit more the experimental structure. So as you can see from this model, we don't get the exact loop conformation, but we get something that kind of resembles the experimental structures. And that is, of course, much better than the original AlphaFold conformation. So we decided to benchmark this AlphaFlow algorithm. We generated 1,000 models for each heavy chain of an antibody. And we saw that with AlphaFlow, you can always recover some H3 loop with an RMSD lower than 3. So something you can actually use for any downstream task. And here you can see the distribution. On the left, AlphaFold 2, its ensemble contains only 25 models. So yeah, it's a bit of an unfair comparison. But on the right, you can see how AlphaFlow can always recover good models in its sample. But of course, now the complicated task is to fish out the good models out of this sample. So how to recognize the good models in the AlphaFlow sample. And here the PLDDT that is provided by AlphaFlow is not really predictive. There is no real correlation with the experimental structure. And to solve this riddle, we developed this workflow that is here schematically illustrated. Basically, in this protocol, you start with an antibody for which the H3 loop is predicted with quite a low confidence by AlphaFold. So in that case, you do an H3 loop focused AlphaFlow diffusion sampling, where you sample hundreds of possible conformations of H3. And then you perform a complete linkage, maximum entry structural clustering. So we understood that this procedure is optimal for performing structural clustering, so it has to retain the best models in at least one good model in the ensemble. And the resulting ensemble, we call it cluster diffusion ensemble, is very heterogeneous, but also small. It can be like 10 models, something like that. And then with this ensemble, you can do, for example, docking. You can do targeted ad hoc docking to your antigen of interest. And we benchmarked this on 2 different information scenarios in haddock. And we show that using these cluster diffusion ensembles, you're always able to improve over the standard AlphaFold 2 baseline. So here you see that if you look at this plot, the para-AP scenario, if we look at the first bar, the top one bar, cluster diffusion ensembles are able to provide a good answer, an accurate answer, almost 80% of the times, while AlphaFold can only do it like around 30% of them. And also the quality of the models that you are getting is improved a lot with cluster diffusion ensembles. As you can see this from this green bar, that we have a lot of medium quality models with cluster diffusion ensembles, while for AlphaFold, they are very complicated to obtain. And now more like a visual inspection of some success stories of this approach. Here we have the prediction of a monoclonal antibody in complex with a glycoprotein from the hepatitis C virus. And here you can see that with cluster diffusion ensembles, we can get a good model using haddock. And this model is very good. The antigen is predicted in the correct orientation, even though the H3 conformation is not perfect, but it's good enough to allow the docking to place the antigen at the correct interface. So this story is a good way to tackle very challenging antibody systems. So now we have AlphaFold 3. Did it solve everything? Well, of course, it improved a lot the success of antibody antigen complex modeling. They claim in their paper up to 60%. And here, the very good thing about AlphaFold 3 for antibodies is that the importance of the MSA is really de-emphasized, And there is also a diffusion module inside AlphaFold 3 that grants more diversity in the sampled structures. So this is very useful for antibodies, where we saw that the MSA is not really relevant, and diffusion can be important to have more diversity. But on our small dataset of very difficult cases, AlphaFold 3 actually helped only in 1 out of 9 cases, and it reached an H3 RMSD of 2 angstrom, so not really an impressive value. So did it solve everything? No, it's a major step forward for sure, yes. So now I think I will spend the last minutes of this webinar to discuss a bit the field of nanobody modeling. And first, I want to start by saying that this is the work from Miguel Sanchez Marin, master students with me in the computational structural biology lab. And in his work, Miguel inspected the differences between nanobodies and antibodies. And a nanobody is what is called a minimal antigen binding molecule. If you look at the structure of the standard antibody, standard immunoglobulin, you can see that it has a lot of domains. It's 12 domains, and a lot of them are conserved. It's only the 2 variable domains of the heavy and the light chains that are actually binding the antigen, and that have some diversity. In camelids, basically the light chain didn't develop for some evolutionary reason, and the smaller antigen binding unit here is what is called the nanobody. And the fact that the nanobody is so small is very useful for applications in which you have to really optimize for solubility, for example. And also to mention, nanobodies are able to retain very similar properties with respect to antibodies in terms of affinity and specificity. But is the sequence of an antibody really different from the sequence of the variable domain of the heavy chain? It's similar, but it's not the same. The first CDR1 loop is a bit longer in nanobodies, and the CDR3 loop is much longer. So we have, on average, many more amino acids in the nanobodies CDR3. And this is a sketch of the structure of a nanobody. And here what Miguel found out is that since the light chain is missing in this case, the framework residues are more relevant and are actually much more often than in antibodies. And here again, CDR3 is the main actor in antigen binding, even with a more prominent role with respect to antibodies. So CDR3 is really the main character in while binding to the antigen. Here you see an example of this binding, where H3 is really like playing the most important part, creating the majority of the contacts with the antigen, with the other 2 loops just giving minor contributions to the binding. What Miguel found out also is that we can define 2 different clusters of binding modes for nanobodies. So 2 different clusters of amino acids that on the nanobodies are used to contact the antigen. So Miguel here took all the available nanobody antigen complexes, and he found out that we can divide them roughly in 2 groups. Basically, in the first one, we see how the nanobody is contacting the antigen using only the CDRs, only the variable loops. So the CDR3 here on the right is always present, while the other 2 CDRs are most of the times there. And then in a second representation that takes into account like almost 70% of the structure, the distribution is much more blurred. So you have still a lot of the CDR loop residues are at the interface most of the times, but then a lot of other residues appear at the interface. So there is a more cooperative binding effect. And here, Miguel set up a nanobody modeling benchmarking study in which basically we benchmark different methods to predict the structure of an antibody. And of course, we also benchmarked the complex prediction with AlphaFold. And at the end, we used the predicted nanobodies and predicted or experimentally solved antigens to build an ad hoc benchmark. And an important thing about this benchmark set is that once again, we removed all the complexes that were redundant with a training set of AlphaFold and related methods. So only the most recent nanobody antigen complexes were considered. If we look at the quality of the nanobody ensembles, we can see that nanobodies are actually challenging, probably even more challenging than antibodies. And all the tested methods failed to basically give an accurate description of the CDR3 loop for like 20-25% of the cases. But again, if you combine different methods in an ensemble, you see how the chances of getting a reasonable model are actually increasing. And so here we found out that if you combine AlphaFold multimer predictions and immune builder prediction, basically only 10% of the times you have a model that is very far from the experimental structure. And surprisingly, AlphaFlow was not really helping in this situation. So the sampling that it was providing was not really improving over the modeling provided by Immune Builder and AlphaFold multimer. And when we look at the docking, Miguel added a new scenario, a mutagenesis-like scenario, in which basically we assume that the user knows two residues on the epitope, for example, extracted with mutagenesis, and of course, no information is provided about the nanobody, so we use only the CDR loops to perform the docking. And here are the results of the benchmarking study, much lower than for antibodies. So nanobodies are more challenging. And here I added an AlphaFold 3 performance, although AlphaFold 3 was only applied to a fraction of the dataset, so the fraction on which it was not trained on. And we can see that when the interface information is very good, ad hoc is really outperforming AlphaFold. But when the interface information is just partial, it's performing on par with AlphaFold 3 and much better than AlphaFold 2. But when you see these numbers, you shouldn't think that, okay, since AlphaFold is having more or less the same performance as, so when AlphaFold and ad hoc have the same performances, it means that they can produce a good model in different cases. And that's why combining the two of them together to have a clearer picture is typically a good strategy. I want to show you just a case in which AlphaFold 3 here is producing a very bad model. Here the correct antibody and nanobody antigen complex is in gray, while AlphaFold 3 generates the red one. And using ad hoc with mutagenesis-like restraints, we get a good model of the complex. So something that you can use for example, for antibody design studies. So to sum up with antibodies in this post-AlphaFold era, really AlphaFold really revolutionized the field of antibody and nanobody modeling, but there are still challenges in the complex and H2 loop prediction. And in some cases, when the H2 loop is very challenging to model, it might be useful to force AlphaFold to explore in a wider manner the conformational space of this loop. And for now, we can say that combining AI-based methods such as AlphaFold with more classical physics and chemistry-based approaches with experimental data is still very useful for gaining unique structural insights of the system, of the antibody antigen system. And what I can say as a perspective is that, especially if you follow the works of the Baker Lab in the last year, we can see that now the structural methods for predicting antibodies and nanobodies are so powerful that a fully computational in silico antibody design protocol is really a feasible option. So this can save a lot of time in experimental work. And with this, I would like to thank you for your attention, and would like to thank a lot my supervisor Alexander Bonvan, and also Xiao Tong and Miguel, and all the other members of the computational structural biology group at Utrecht University, and of course the Bioexcel for inviting me here. Happy to take any questions.
Thank you very much, Marco. Do you hear me? Yes.
Yes.
Okay, great. So we have a question, and that is, do you have any comparison for prediction accuracy with respect to RF diffusion?
Okay, maybe I can stop sharing for this. No, we don't use RF diffusion because here we deal only with antibodies that are known in sequence. So we don't generate the backbone with RF diffusion. So no, we don't have any comparison about that.
Okay, thank you. We had another question. That is a long question, so I will read it. I don't know if you can also read it, but I will anyway read for everybody. So interesting talk. Do you think ADOC3 is able to predict antibody protein complex of different antibody for the same protein? So this is the first part, and we say we know from the wet lab data we have antibody that bind different epitopes of the same protein, but do not know the region of any key residue at this time. Do you think ADOC3 will be able to distinguish and predict binding location for the different antibodies? The protein is very large, around 1,700 residues.
So this is a tough problem. If you had at least one vague definition of at least one epitope, then this antibody screening would be feasible with a haddock. But without any information about the epitope, what I would suggest would be to try AlphaFold first, so that maybe AlphaFold will give you a hit, and then maybe you can optimize over this hit, because ADOC is not really meant to be used in absence of any information about the epitope. So for that, more like ab initio end-to-end methods such as AlphaFold are going to perform better.
Okay. So then I was trying to find, I thought I saw one person. Okay, I will try now. We have a question from Manuel Orozco. I will allow you to talk if you want to ask your question yourself. Please go ahead, Manuel. We don't hear you. We
Can you
saw
hear me
that.
now?
Yes, we can hear
Okay,
you.
perfect.
Please.
Yeah. Thank you. Very nice presentation. I was just wondering if it's possible to use or feasible to use MD simulations as a means to sample the conformations of the H3 loop and use that as an ensemble for input for the docking calculations.
That's a very good question. In short, yes, it's very feasible. If I elaborate a bit more on that, well, it is known that the free energy barriers between different conformations of H3 are not easy to be overcome. So even the MD's based sampling is going to take a while. But we have a workflow within Bioexcel in which we are actually combining GROMACS and haddock, in which GROMACS is actually sampling alternative conformations of H3, and then those conformations are clustered and used within haddock. So I would say that the approach that I highlighted is more like complementary to that. It's another way of doing the same thing. One is based on MD, and the other one is based on a diffusion model on top of AlphaFold, but both are fine in principle. The MD one is going to take more time.
Okay, thank you very much. So now we have a question of Arun. Arun, I will allow you to speak. Now you should be able to speak. If you unmute yourself. Okay, maybe Arun. Okay, No, maybe it does not have the microphone. So I will read the question. Can you suggest method for prediction of dia bodies?
Dual bodies, I guess.
Yes, yeah, it could be dual bodies. Yeah, you see the question, I guess. Yeah.
Yes, let me see. Yeah, I guess it's
Yeah,
dual bodies.
yes, I guess so.
So
yeah, we don't have.
it's a tough question. I would start by using the single hypervariable regions. So without taking the dual body itself. So I would start by predicting the interaction between each arm of the Fab and the antigen, and then once you have the two models, so the model of the first body and the model of the second body, then you can try to combine them together using, for example, homology modeling on the template, or even haddock. But I would really start with the prediction of the single variable domain attached to the antigen. That's the challenging problem. After that, you should be able to find out how to build a full model of the dual body interaction.
Okay, thank you very much. Now I will allow the Caris to speak. If you want to tell us your question, please go ahead.
Hello. So also just wanted to ask more like technical side. Regarding that comparison you had between the predictions for the binding with and without knowledge about the interface. When you refer to interface information, is this basically just saying we know where on the antigen it binds, or is this referring to more detailed information like, say, it's binding through hydrophobic interactions versus hydrogen bonding? Yeah, or is it just basically just in general the location of where it's binding that helps improve the predictions?
It's it's a great question. Haddock is designed to take into account any type of experimental information. So the more accurate is the information you have, the more accurate will be the prediction. So in this case, I was describing a very coarse information that is simply a list of amino acids that you think might be in contact with the antibody. So nothing more than just a list, just a patch on the antigen side. But of course, if you know more about that, for example, this doesn't happen very often, but if you know, for example, a single contact, you can also specify that in Haddock. Haddock can take any type of experimental information in input and use that to basically drive the modeling. That's its major power.
Thank you. Now we have a question from Alexandro. I will try to unmute him. Yeah, Alexandro, if you want to ask, you can speak. I guess he cannot unmute himself, so I will read the person will say. So yes, excellent. How apply your method for studying protein ligand complex?
Yeah, that's another problem, but we have a lot of tutorials about how to use Haddock for protein ligand prediction. Again, it's the more information you have about the binding site of the ligand, the better the modeling will be. But if you check our website, so bombenlab.org, you will see a lot of tutorials about how to use different ligand information to perform basically protein ligand modeling.
Okay, then we have a question from an anonymous attendee. In future, do you think AlphaFold-like methods in vacuum can come close to MD simulation in solvent? Following up on the two method, for example, of CDR H3 loop conformational generation.
So I got the first one.
So it's
blah 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 can come close to MD simulation since solvent. Following up on the two methods for yeah,
I think what you presented.
So I think the future is going to be about the development of a machine learning method that is able to generate MD-like conformations at a fraction of the cost of MD. That's the future, and this is something we are already seeing with the release, for example, of a bioemulator or even AlphaFlow, and yeah, I think we are getting there. But like as with everything else with machine learning, you know, the machine learning can only sample things that kind of resemble what has been observed before. So for very challenging systems, for systems for which we have no data, I still think that in order to replace MD, that's going to take a while.
Okay, so now I still take one or two questions. So the next question is from Vesel Vold. I allowed you to speak, if you can.
Thank you for the presentation. Could you please share some details about implementation of HEADDOC? I see that it uses Python. What about its performance in comparison with AfterDoc? I'm sorry, with AlphaFolds, for example?
Yes, well, it uses Python as a wrapper. So in the compute-intensive routines are written in a variant of Fortran in CNS, and that is very fast. So everything that requires a lot of computing time is basically super optimized. But yeah, of course, AlphaFold is going to be faster most of the times, depending on the length of the proteins, basically. So AlphaFold is very fast for like small proteins, but its computational cost grows a lot when you have bigger proteins. While for HADOC, that doesn't make a big difference. But yeah, a standard HADOC workflow will take 1 hour and 1.5 hours, 2 hours, with using at least 10 cores on a CPU. So probably you can run AlphaFold faster with that. But if you want to sample more seed with AlphaFold, that's going to take a while. AlphaFold 3 is now faster, but yeah, it's not free for commercial use, and it has all its problems. So HADOC is a feasible solution. It's not cheap computationally, but it's not that it's taking days to run.
Okay, thank you.
Thank you very much, Marco. I take the last question and then I close, because that we are otherwise over time, and I will share a little. So one of the last question that I will take is, will it be possible to use in a glycan interaction in the future? Glycan, sorry.
We have done a lot of efforts to understand the interactions between glycans and antibodies and proteins in general, and well, wait, did I say AlphaFold or HADOC for glycan interaction?
It didn't specify. Will it be possible to use in glycan interaction? I guess HADOC.
Okay.
This is my
for
assumption.
AlphaFold, yes, it's already for HADOC. It's already doable. We already have a lot of basically all possible glycans as part of our list of supported molecules. So you can already do it. We wrote a paper about this in last year. So check it out. Of course, you need some information about the interaction, about the location of the interaction between the glycan and the protein. And with HADOC, you can also model like covalent interactions between like pure glycosylation of the protein. So that's very easy, and you can check out our tutorials on the website.
Okay, thank you very much. So I just want to close by announcing the following up activity from Bioexcel. That will be a workshop that we run in Sofia, the 21st and 22nd May. Please hurry up if you're interested to attend. And HADOC will be also HADOC 3. There will be exactly a tutorial on HADOC 3 in that workshop. So then we will have the next occasion. So the next webinar will be the 13th of May, and we will speak about QMMM, and soon will come out the announcement. So I would like to thank everybody for the active participation, Marco for the wonderful presentation, and all the attendees for a lot of questions. We have questions that are not yet answered, so we will always suggest to talk those questions in Ask Bioexcel forum under ad hoc, and Marco will be happy to answer those questions. Thank you very much for attending. See you
Thanks a
next
lot, everyone.
occasion.
Thanks a lot, Alessandra.